Assalamu alaikum. The topic is cytoskeleton. What does the word cytoskeleton means? It means the cyto is the word used for cell and skeleton is the framework of that cell. So whatever the framework of the cell, it is the basic one network of the fibers deeper to the plasma membrane of the cell that maintains the shape of the cell and is responsible for the motility of the cells and is also responsible for the intracytoplasmic movement of the organelles and the vesicles. Now what does this cytoskeleton comprises of? This is basically protein in nature, and three type of the fibers they are and structures they are making this cytoskeleton. You can easily appreciate these structures over here. The first one is a smaller one filament, that is microfilament, and usually within the cells, within the eukaryotic cells, it's the actin filament which is labeled as microfilament. The second one structure, this is the intermediate filament. This is the second part of the cytoskeleton. Similar to the bones within the body of the human, there is a skeleton or a framework within the cell, and this is proteinaceous in nature, and the structures which are making it, this is the first one, the microfilaments, second one the intermediate filaments, and third one is microtubules. These are smaller tube-like structures, microtubules. These three structures, three type of the structures, they are making this cytoskeleton or the basic framework of the cells. Now starting from the smallest one, that is starting from the microfilaments, they are called as micro just because of their size or the diameter, and they are basically made up of actin subunits, a protein, a smaller protein, actin, that makes these fine strands. So its microfilaments is basically the actin filaments, and they are having the diameter of about 7 nanometer in 7 nanometer, and the range lie between 5 to 8 nanometers, but they are usually about this 7 nanometers, and each of these, they are made up of subunits which are called as the actin subunits. These subunits, they are joined together to form and they are twisted upon each other to form a twisted string, that is the basic structure of the actin filaments. Now what are the factors that are responsible for the stability of the or for this formation of these filaments? They are basically the calcium ions and the ATP molecule. In the presence of the calcium ions and the ATP molecules, these actin filaments, these actin subunits, they are joined together to form the filaments, and they are maximally stabilized within the presence of these two things. Now the structural and the biochemical studies, they are telling us that they are of different types. However, the basic types are two in nature. The first one is globular actin. The globular actin is the free actin that is not in the filamentous form and is present within the cytoplasm. The second one is F-actin, which has taken the shape of the filaments. Like the same globular actins, when joined together to form the filaments, they are called as F-actins. So actin is present in actin filaments, or the actin is present in two forms, the G-actin form, globular actin form, and within the filamentous form. Now how does this actin filament this is going to assemble? At first, the globular filaments, they are present within the globular actin. This is present within the cytoplasm. This is a protein, and this is freely present within the cytoplasm. But whenever there is an increase amount of the energy molecule, that is the ATP, adenosine triphosphate, this is there within the cell, these adenosine triphosphate molecules, they get bind to these G-actin proteins. Now these G-actin proteins, they become hyperactive. active with the presence of the adenosine triphosphate or ATP molecules. When they are becoming highly active, they are fusing with each other to form dimers or trimers. The dimers or trimers, they are there, and whenever there is dimers or trimers and in the presence of the ATP, they are going to assemble upon each other to form the filaments. You can easily see this filament. This is basically the red one is the G-actin. Along with that, this ATP molecule, the purple color molecule, this has been incorporated into it, and all of these, they have this one. Now, as I have told you before, that these actin or globulin subunits, they are polarized structures. This is a polarized structure. You can see the dark color over here and the lighter color on the top side. This is just to show you that it's a polarized one. It has one of the end is negative and the other end is positively charged. So there are two, this is polarized one molecule, and it has two portions. One is negative subunit and the other one is negative part and the other one is positive part. If you look at here, the assembly is always on the positive side. The negative side is this one, where this is the lower one side, and towards the positive side there is polymerization or the addition of the this activated globular actin. This is on the positive side or plus side. So we always used to say that polymerization is on the positive side, and the depolymerization or disassembly, this is towards the negative side. What is this disassembly? Whenever there is the use of the energy molecule in making this filament, the lower one portion, at the lower side, this energy molecule ATP has been hydrolyzed into the ADP. Now, from where this enzymatic activity is coming? This is the enzymatic activity within this subunit molecule, that is within the actin molecule or actin subunit. It's a protein, it's an enzyme, it has the hydrolyzing capability for ATP molecule. So whenever there is utilization of this ATP molecule and the energy has been removed from it, it has been converted into adenosine diphosphate. Like triphosphate, one of the phosphate has been used to give up the energy, and the rest of the two phosphates, they are still joined together. Now this globular one protein, this after losing this one phosphate, this is now ADP bound, and this is in the lower one portion, or you can say it is in the back in the resting state rather than an activated state. So at this site, this is going to degrade and this is going to depolymerize. So polymerization is always on the positive side or the plus side, and depolymerization is always on the negative side, that is the minus side of the actin filaments. Now within the cell, they may be organized in different forms. In different cells, they may be present in different types or different appearance, gives a different appearance. Like at the most one, they are present in the skeletal muscles, but they are present in the skeletal muscles along with the thick filament. This is the myosin filament, the thick filament, about 15 nanometer in size, and this smaller one, this is the spirally acting, the actin filament, or spirally twisting these actin filaments. Now within the skeletal muscles, these actin and myosin, they are present in paracrystalline array side to side with each other. And if you look at here, these are the heads of the multiple myosin filaments. The myosin filament, this is the single one unit, and this myosin filament has two globular heads, and you can easily look into here, this thick filament carries multiple myosin filaments. So multiple myosin filament join together to give us a thick myosin filament, which is visible over here, and this arrangement is present along along with the actin within the skeletal muscles. And usually the actin filaments is richly present within the skeletal muscles. Another one form in which they are present within the cell, this is an unorganized form of the filaments. Like filaments are there, but they are not organized in a specific way. They are present freely within the cytoplasm. The third one form where they are present within the cell in an organized form, this is by forming the cell cortex. What is cell cortex? This is basically the actin filament. They are gathering together deeper to the plasma membrane, forming a sheath around the cytoplasm. This sheath is called as cell cortex. So the cell cortex, this is immediately lying deeper to the plasma lemma, and this is the one which is responsible for the membrane activities of the cell, like exocytosis or the endocytosis. Whenever the cell has to the membrane has to bulge out to give the pseudopore, this is there, and this is the one which is basically raising at first and then taking the plasma membrane along with that. And second one, membrane activity, this is the migratory activity of the cells. The cell has to move by amoeboid movement or by any other way. This is basically the cytoskeleton or the cell cortical cytoskeleton actin filament, which is raising that foot or the appendage of the cell to move that towards the site. So the two forms, they are there in the skeletal muscles and then in the form of the cell cortex, and the third one is an unorganized free form, and the fourth one is there is actin filament involved in the cytokinesis. What is cytokinesis? Cytokinesis is the cleavage of the cells into two parts, or cytocell kinesis breakdown. So this is basically the end of the mitotic activity within the cell. That's one cell turns into two, and whenever there is a breakup of the cells into two, there is involvement of the actin filaments. These actin filaments, along with the smaller protein subunits myosin, they are making a bursting appearance around the site of constriction, and when they are making this appearance so that they can constrict the cells, or whenever there will be contraction of these things, these two proteins, they make the cells to contract at this site. Ultimately the cell is basically break up into one. So it is involved in the cytokinesis. The fifth one form where it is present within the cell, this is the way where it is responsible for the movement of the or it is associated with the movement of different vesicles and the granules within the cytoplasm. This process is called as cytoplasmic streaming. If you look at here, this is the inner side of the cytoplasm, and so the actin filament, this is making a framework over here. Now if one organelle, this is bound over here, this actin filament, it will contract and it can pull that organelle to the other side. So movement of the cytoplasm or movement of the vesicles within the cytoplasm, this is another one function performed by the actin filament. You can easily see over here, this is the once again the proteins, and along with that proteins, the actin filaments, they are there, and these actin filaments, they are now, this is involved in the focal adhesion. Like some organelle may be present over here and it may bound to the actin filament on one side, and then whenever there is contraction, they are pulled over here. So this is called as, this is the same thing that is visible over here in the broader aspect of this thing is visible in this lower one picture. This is called as focal adhesion. So actin filament is also involved in focal adhesion. The maximally stable actin filament is that one which is present within the muscles. The other one cells, it do carry the actin filament, but they are readily dissociate and readily reassemble. Why they are most stable within the muscles? Because the muscles have an increased concentration of the calcium ions, and secondly there is an increased energy protein or energy molecules over there, so that in the presence of the calcium and the ATP they are maximally stable. Second thing which is required, third thing which is required along with that, this is the actin binding proteins. For their stability, the calcium and ATP is required, but for their stability, another one thing which is required, this is the actin binding proteins. Otherwise, if the actin binding proteins are not there, they are there, but they cannot perform their function as collective one. So what are the actin binding proteins? They are different type of the proteins present or spread throughout the cells. The first one is the bundling protein. They are responsible for bundling of the actin filaments. Actin filaments is present within the cell in the monomeric form. Whenever they are these monomers, they are joining together, there must be some binding force between these monomers. So these monomers, they have been bundled up in parallel bundles by the presence of the or along with the presence of the bundling proteins. An example of it, this is within the microvillus, the actin filament, they are present within the microfilament, and the bundling proteins over there, they were the fascin and the fibrin. Second one, protein, actin binding protein, this is of another type, that is the severing one, actin filament severing one. Severing one means the cuts the protein at one side so that they are not lengthening and lengthening. The severing one breaks them up into smaller subunits. So the severing protein is the one which is not letting the actin filaments to polymerize and moving and coming out of the cell, rather than it is breaking it up into smaller subunits. And the example of the actin by these severing proteins, this is the gelsolin. It is responsible for making the gel like actin filaments into the liquid form. So the third one is actin capping protein. If you look at here, this one is the capping protein, or you can say, you can easily identify these caps over here or these caps over here. They are the capping proteins. The tropomodulin is an example. They cap up the filament so that they are no more lengthening. The another one type, this is the actin cross-linking protein. Like bundling proteins, they are making the bundles of the actin filaments in parallel forms. However, whenever there is cross-linking required, the another one protein having the two limbs or in the form of V, or you can say the Y form, they are present, and they make the bundles they makes the crisscrossing of the bundles. In the Y form they are, or in the V form they are here. The two ends of the V or Y, they are attached to the actin filaments, so that they are bundled in the cross-linking forms. Example is within the cytoskeleton of the erythrocytes, the spectrin protein, this is there, which is responsible for maintaining the shape of the erythrocytes. Another one protein, actin binding protein, which has been quoted before, this is the motor proteins, that is the myosin protein. This belongs to the myosin family, and multiple myosin proteins, they are there within the cells, and they are the one which is the motor one. They have the capability of hydrolyzing the ATP to provide the energy for the movements. So all of these, they are the actin binding proteins. Now the second one portion within the cytoskeleton. This is the intermediate filaments. Intermediate filaments are called as intermediate because they are their diameter is ranging between the microfilaments and the microtubules. So this is the second component of the cytoskeleton, and its average diameter is about 10 to 12 nanometer. And several proteins, this is once again proteinaceous in nature, and several different type of proteins, they have been isolated, forming these intermediate filaments. But these intermediate filaments, they have some specific characteristic features different from that of the microfilaments and microtubules, which are making them purely structural in nature. Now, what are these specific features which makes them unique from that of the both of the microfilaments and microtubules? They are, first of all, they are not the polymerized one, not the polar one. So their non-polar nature makes them purely structural in nature. Second one, they have a considerable diversity and tissue specificity. What does it means? They are having the specific tissue specific nature. Supposedly one of the filaments, they are present in the all of the structures derived from the mesenchyme, so they may be found in any sort of mesenchymal derived structure. However, its quantity can be variable in that, but they are surely present over there. So this thing is important in recognizing the, or in whenever we are doing the histopathology of the tumor cells. This thing is very important in diagnosing the cancers, whether this is of the primary origin, or they are of the they are the mets and coming from the other sites. Like supposedly the site of the all of the epithelial origin tumors, they have the cytokeratin intermediate filament within them. Now if the primary met is from the epithelium, this must have this intermediate filament. If it is present deeper within the tumor has reached to any other site, or deeper within the muscular layer of the any site, even then whenever there is histopathology of that tumor, we can easily identify that its origin is from the epithelial site. Now it has moved over here. So this is why how it is the having the diversity and the tissue specificity. Now whenever I'm saying it is a non-polar filament, or I have, so it cannot be easily disappear or cannot be easily reform. Like polymerization is not so actively present within the intermediate filaments as is present within the microfilaments and microtubules. This is the reason why it is making it structurally stable one structures, and they are playing a role in the structural stability of the cells by making the links with the and by making a continuum or a network within the cell cytoplasm. So this is basically involved in the in bearing the tensile strengths and the shearing strength, giving the strength of the cell against the shearing forces. So basically the intermediate filaments, they are structurally stable filaments, and they are giving the structural stability to the cells. Now how they are formed? Initially they are having the monomeric appearance. These monomers, they are joined together to form dimers. Now these dimers, they are bounding together to form the tetramers, and these tetramers, they are joining together to form the octamers. So each of the filaments, they are not having the single monomeric structure, they have the octameric appearance, and or more than that, multiple octamers, they are joined together to form the intermediate filament. That's why its thickness is more than that of the actin filaments, which is only the dimers. So what are the different classes of these intermediate filaments? There are usually the four classes. If we break them up broadly, they are three classes. They are present within the cytoplasm, and fourth type, this is only specific for the nucleus. Now the nuclear intermediate filament, this is called as nuclear lamin. Nuclear lamin forms a lamina or a network deeper to the nuclear membrane. This is the movement of the nuclear membrane from here, and whenever the nuclear membrane this has been removed from here, deeper to that this is a network or a sieve-like structure. This is called as the made up of the nuclear lamins. Now these nuclear lamins, they are the one which are intermediate filaments, and they are specifically present only within the nucleus. So from where the source is coming, this is visible from this histopathology of the nucleus, this one, the intermediate filaments. Other types, they are present within the cytoplasm. What are these? The first one, this is present within the epithelial cells, and they are made up of keratin or cytokeratin. The cytokeratin filaments, they are present in different fibrils forms within the epithelium, and their maximum amount is present within the epithelium of the skin, because it is having the characteristic feature of defense against the abrasions and the loss of water and the heat. However, its amount, this is variable in the other epithelial cells. Supposedly the epithelial cells of the intestine, this is not exposed to the such sort of heat and the temperature, so, or the external environment. So this is having the cytokeratin, but its amount is less as compared to the stratified squamous keratinized epithelium of the skin. So its amount is variable according to its position, but it is always present within any epithelium of the body. So wherever there is epithelium, the intermediate filament cytokeratin, this is there. Second one type, this is mesenchymal origin. What is mesenchyme? Mesenchyme is an embryonic tissue, the intermediate tissue between the between the three germ layers. Whenever a germ layer cell, this is converted to the other one, this the intermediate interwoven tissue, this is called as mesenchyme. So from that mesenchyme, we have the development of the connective tissue cells, we have the development of the muscle cells, we have the development of the glial or the supporting cell, and the intermediate filaments which are mesenchymal in origin. The examples are vimentin. This is present, this is a single protein which may polymerize with the other one. And the second one, this is the desmin, specifically found in the smooth muscles. However, it may be present in the this is present also in the lines of the Z lines of the skeletal and the cardiac muscles, but specifically they are present within the smooth muscles. The third one, which is also once again the mesenchymal origin, this is the glial fibrillary acidic proteins. The glial cells are the supporting cells. They are not the neuron cells. They are a part of the nervous system, but they are basically the supporting cells. So on the nutritive and the mechanical support of the neurons, this is by the glial cells, and these glial cells, they have the glial fibrillary acidic proteins. So this, all of these, they are the mesenchymal origin, having the mesenchymal origin. So mesenchymal proteins, they are the intermediate filaments, they are these one. And the third one, this is once again present within the cytoplasm. This class is specifically the neurofilaments. They are of the three types, and they have different characteristic features. They are present only in the nerve cells. So these four basic types of the intermediate filaments, they are present in the human body. Next one is microtubules. As the name is showing, this is a smaller tubule. Tubule mean inner hollow structure. These are tube-like structure with the narrow, with the inner core and the inner tube-like cavity, and inner hollow core is about 14 nanometer in diameter. However, the outer diameter, this is the 24 nanometer, and if we count the each one subunit of it, this is having the diameter of 5 nanometer. So the whole one wall, this is 5 nanometer thick, and on the inner side the central diameter of the hollow cavity, this is about 14 nanometer. However, the external diameter of the microtubules, this is the 24 nanometer, and they are of the variable lengths, and according to different sites they may have the arms or the bridges. Now, what are their functions? They are basically involved in the intracellular vesicular transport and intracellular organelle transport. They are making a railway road track within the cell, so that the upon with this railway track, the vesicles and the organelles, they move along with the via the attachment of the proteins. So they are performing they are giving a track within the cytoplasm for the movement of the vesicles and the movement of the organelles within the cytoplasm. Secondly, they are microtubules. They are basically the basic structure of the cilia or the flagella, and these two cilia and the flagella, they are basically the mobile parts of the cell. So their movement is because of their inner microtubule presence, or the inner structure of the microtubules. Third one, they are present in the cell in the form of mitotic spindles. So attachment of the chromosomes to the mitotic spindles and their movement, this is because of the microtubules. Similarly, the elongation and the movement of the cells and the maintenance of its shape, in which this is also playing its role. Now, what is microtubule structure? The basic subunit which is forming these microtubules, this is a protein tubulin. Tubulin has alpha tubulin. There are two types of the tubulins within the cell, alpha tubulin and the beta tubulin. Once again, they are the polarized structure, that they have the two poles, one end is negative and the other one is positive. So the two tubulin proteins, the alpha and the beta types, they are forming they are joined with each other and they are forming a heterodimer. So this heterodimer, this is polymerizing to give us this protofilament of the tubulin. The single one is over here, visible over here, and these single one, they are making a filament. These filaments, they are called as protofilaments. About 13 protofilaments, they are joined together to give us this microtubule, and they are present in the form of in one complete turn of the spirals. So there are about 13 subunits, the 13 subunit protofilaments joining together to give us a microtubule. Now, why or when these microtubules, they are present or they are formed? They are basically formed once again in the presence of the calcium ions, in the presence of the ATP molecules, and in the presence of the microtubule associated proteins, but under the direction of organizing centers. So there must be an additional one microtubule organizing center within the cell, so that these microtubules, they are polymerizing along with the presence of the increased calcium and the energy molecules. Now, what are these microtubule organizing centers? We have basically the basal bodies and the centrosomes, which are the basic microtubule organizing centers. If we look at the microtubules, these are the microtubules within the cell. Under the microscope, they are visible like so. the two ends of the tubulin protofilaments, they are visible, and with the central cavity over here. This is once again these tubules visible over here and under the microscopes. Now what are the how these are there, and how they are being polymerized. Once again, they are polymerizing in the same way that they have the polymerization at the positive end, and depolymerization is along the negative side. So polymerization is always on the positive side or the plus end of the microtubules. Now, if we look at the microtubule organizing centers, at first they have the basic structure of the centrioles. What are these centrioles? Centriole is one of the organelles. These are cylindrical structures about 0.15 micrometer diameter, and their length is variable. In each of these, they have basically a specific arrangement of the microtubules. The microtubules, they are present in the form of the triplets. Like triplets are joined at the periphery. 9 triplets joined together at the periphery to give us a basic structure of the centrioles. And this centriole is the one which is present within the if you look at here, this is present within the centrosomes. Two centrioles are present at right angle to each other to give us the centrosome. Centrosome, along with this centrioles, it is also carrying the granular extracellular area matrix, or the different extracellular matrix carrying the rich amount of the proteins which are required for the formation of the tubules, or the which are required for the formation of the microtubule or the mitotic spindles. So they are there. The similar sort of structure, this is also present within the basal body. Each basal body is present at the lower side of the cilia, and if you look at the cilia has the structure of 9 doublets. Doublets are there at the periphery. However, along with the doublets, there are two singlets present in the center, and each of these, the singlet one, this is also involved in this is also there in the form of having a sheath, and the triplets and the doublet at the periphery, they are also having the sheaths. So the cilia is having this sort of structure, and at the base of each cilia there is the organizing center, which is the basal body. So centrosome and the basal bodies, these are the microtubule organizing centers, and the cilia, or the other one thing, the mitotic spindles, these are the structures which have the microtubule, or the microtubule structures. Now, how does the microtubule associated proteins, they are playing their role? The organizing center, it is giving the mitotic spindles, but mitotic spindles they can join with the chromosomes only in the presence of the proteins, the microtubule associated protein kinetochore. If the kinetochore is not there, the mitotic spindle will come back once again in the form of a tongue coming out of a salamander. It comes back to its center, but in the presence of the microtubule associated protein, it may join with that, and on the other side of the kinetochore, the chromosome is attached. So basically, the chromosomes are not directly attached to the mitotic spindles, they are attached along with the presence of the microtubule associated proteins. So this is how the presence of the proteins along with the centers, this is there within the cells. Now, what is how the anti-cancerous drugs, they play their role in making the halting the growth of the cells? All of the mitotic cells, they have the centrosomes, and we have studied it in the earlier classes that there is duplication of the centrosomes within the S phase of the cell cycle, and after that they direct the plane where the cleavage of the cell this is going on, and from these sides the mitotic spindles they are going to form. Now if the mitotic spindles they are not formed, the chromosome is not going to bind with that, and the cell this is not going to be duplicated. So anti-cancerous drugs like colchicine and the Taxol, they are going to form, they are going to be attached to the tubulin molecules of the microtubules, so that there is deformity of the formation of the microtubules. In this way the mitosis of the cells within the cancers, they have been halted by the anti-cancer drugs. So this is the whole thing with the related to the cytoskeleton. Now cytoskeleton, so once again taking in view the basic function, the basic parts and the functions of the cytoskeleton. What are these? The first one is microfilament. This is basically involved in maintaining the shape of the cell. It is going to change the shape, and it is also involved in the contraction and the cytoplasmic streaming and the cellular motion. The intermediate filament, this is the one which is giving the resistance to the structural support to the cell, and it is the one which resists the tensile strength. And the microtubules, these are the hollow dynamic structures, and they move the chromosomes and they move the organelles from one side to the other side. So this is the whole thing regarding the cytoskeleton. Thank you very much.